Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Let's talk something aside from science. Today we will talk about the connecting story of generations. Why I choose this topic, what I will discuss. It's an interesting story and I will try to convey in a kind of story manner or storytelling presentation. So let's see. The story uh, of connecting generation belongs to actually the Nobel Prize in Chemistry for the year 2024. We got three persons for this Nobel Prize, and two are sharing uh, Professor Hasabi and Professor Jumper are sharing half of the prize with Professor David Becker. Professor David Becker and Jumper, they are Americans, while Hasabi is from UK or England. So, but why I choose this topic that the connecting story of generation, and also why I am covering only the Nobel Prize winner in chemistry, because all of these three, they are from different background, somehow from different background, but their work is connecting to each other. And for them, they received a Nobel Prize. And if you will listen this story, it's indeed an interesting one. So first of all, let's talk about Professor David Becker. He is a PhD in biochemistry in 1989. That means he is a senior professor, and I will keep him in generation first. The other two, Professor Hasabi and also John Jumper, in generation 2, and David Baker will take the generation 1. You can see from here he is kind of a senior guy. Okay, he is basically a biochemist. I will call him a computational biochemist. But since ages, or I mean from the time he joined university and all these things, his main focus was on protein structure predictions. If you know about something about the bioinformatics or the protein structure prediction, so I will just give you a brief history. Um the first method that was very famous and almost with higher accuracy was homology modeling. And for that method, people were using the program of Soli, or you can say Soli Lab. He is from UK, and the program name is Modeler. Modeler, a lot of people work with Modeler and produced astonishing results. But the limitations of Modeler was that Modeler uses a template structure. Now, where the problem came? The problem came because we do not have a template for hard targets. What are hard targets then? For example, you have a gene or you have a protein, you want to predict the structure of that protein, and for that you will perform first a blast search against the PDB, and if you find some another structure from the same family, then the modeler program will use this structure as a template and will predict a new structure for you. But what if you don't have a template structure, then it means homology modeling is not working. And from there, problem came like then how we can predict a structure. And there are many names, but among them, Professor Baker is on top. Why? Because his lab has generated tremendous softwares. I will name it specifically Rosetta, or people will call it, or will maybe you have submitted a job, Robita or TR Rosetta server. And actually, if you see now, they are from Baker's lab. And Baker Lab is situated in the University of Washington, D.C. And in the meanwhile, then if I talk like 2010 and 11, at that time, they had produced or they had generated another state-of-the-art program. Still, it is it is active. Its name is Gremlin. And what Gremlin is doing, Gremlin actually perform coevolutionary analysis between different amino acids, and then the same group, the Baker group, use the information collected from the Gremlin and feed it into the Robita and Rosita and also the TR Rosita. Let me tell you, the TR means transmembrane. So if you are dealing with a transmembrane protein, so I would suggest to use TR Rosita compared to Robita. Okay. And at the same time, there was another server, iTaser. So these all were the kind of competitor. And there was another group, possibly from Finland, and the name of the author was Marcos, and they produced a paper in 2011, I guess. And the paper name was DCA, direct coupling analysis. So actually, the DCA thing was a direct competitor of Gremlin, because both are working almost for the similar tasks to detect the coevolutionary information and then use those information for structure prediction. From this story, what I want to tell you that Professor Becker actually works since long time for protein structure prediction. Okay, but he did not receive the award or the Nobel Prize for the protein structure prediction. But for what then he received the award? This is another story. And then what are the contribution of Hasabi and Jumper? And for what they received the Nobel Prize award? We will discuss. So that's why I kept the title The Story of Generations. Okay. So, Professor Baker, no doubt, he presented state-of-the-art programs, Robita, Rosita, Gremlin, and many other Rosita Fold, etc., etc. But then he shifted his his interest, and they started protein design, or we can also call it de novo protein design, which is now becoming a reality since, I mean, during the COVID time, they had produced some of their design protein in wet lab, and across the globe, different researchers also got success. Still, this field is a new field, but now it's becoming a reality. Okay. What is protein design? Baker and his colleagues or its co-workers, students, postdocs, they thought that why not we make our own sequence and then make fold it or fold that sequence into an active protein and then express that protein in a bacterial system or an eukaryotic system, whatever, and to see whether it will be successful or not. So they got success. And why protein design is important? Because by this technology, you can design a protein of your own choice and of the functions you decide. You can design a peptide as a drug, as an inhibitor, blah, blah, blah. And you can design a complete protein. So let's see, the future is coming and now it's becoming a reality, and possibly in near future you will see um, so many. Maybe we will call it artificial protein or synthetic protein. Okay. Now, the other half of the Nobel Prize is shared by Asabis and John Jumper. That's an interesting part. They got this prize for the protein structure prediction. But let me tell you, Baker got this prize for protein design, not for the protein structure prediction, although he had a long-lasting, he served and he gave too much to the field. But why Hasabi? Hasabi is relatively a junior person compared to the Professor David Baker. So David Baker got his PhD in 1989, and Sabi got his PhD in 2009. So you can guess this is a person from another generation, or I will call kind of a student of Professor Baker. But do you know Hasabi is not, he has nothing to do with the biochemistry or protein stuff. He is basically, or he was a basically video game designer, and he has produced some good games. One of them is a theme park, which he produced in 1994. And he was a good chess player, and according to him, I was motivated from the computer chess, like how computer is playing a chess and how the computer is responding. On the other side, he was really interested in the neurosciences, and he was a computer scientist, a kind of program developer. He has expertise in artificial intelligence, like machine learning, deep neural networks, and all these things. So he got his PhD in neurosciences, and then he created a lab, and its name was DeepMind, and the DeepMind was bought in 2014, and then it became Google's DeepMind. Okay. And Hasabi was also there, and still he is there, a kind of co-founder of Deep Minds and kind of CEO, you can say. Okay, so this is Hasabi's profile. Now, his another colleague, John Jumper, uh, this here is a mistake. He did not produce the theme park, so ignore it. It was just a mistake in the slides. Okay, so he got his PhD in theoretical chemistry in 2017. Now you can guess how much junior he is from what we say from David Baker. Although his BS is, I mean, he has some gaps between the PhD and his BS, so that's why he is just kind of, I will say, kind of the same era with the Hasabi. So that's why I name it uh, Generation 2. Like he is a person from Generation 2. He is also working with Hasabi, and they both are working in deep minds. But what they did, like they received the Nobel Prize, top part of the Nobel Prize. Although I told you that Baker is working in this field of protein structure prediction since long ago, and why they got this Nobel Prize, half of the Nobel Prize, and David Baker got it for protein design. So let's discuss. To understand, you should know about this CASE thing. CASE stands for Critical Assessment of Structure Prediction, and CASE is a kind of meeting. What kind of meeting it is? It gathers scientists from all around the globe who are working in the field of protein structure predictions. And they ask them to come with your softwares, and we will give you a kind of hard target. I mean the hard sequences which are hard to fold or which are hard to predict for structure, and you all will test your program to understand simply, and we will see which program stands the best. Okay, so in and this meeting these meetings are initiated in 1994, and every 2 years these meetings are taking place. If I'm not wrong, in 2022 it was in Turkey. Possibly, I think it was in Turkey 2022 and 3. Okay, so in 2018 there was case meeting number 13, and the whole world heards about a news that there is another program. Its name is AlphaFold, and they almost it's in front of. They produce good results from iTaser and Rosetta. Why I mention iTaser and Rosetta? Because from 2009, I would say 9 and 10 to the 2018 to this era, the whole field was captured or dominated by the Baker Group, Rosetta, Robita, iTaser, all these things. Okay, but for the first time, something new happens, and uh, it was kind of commercial people, because Google is not a university. So that was second part of the news, like Google's DeepMind's program, AlphaFold, and they say that it has produced a bit better results from Rosetta. Now people start asking them to release their code, but they didn't, and it was just became some kind of mystery in the scientific circles that how will it go? Because if it is better than Rosetta and iTaser, so why they are not releasing the code? It's okay, we will not discuss, or we don't have that information that why they didn't, but in 2020, in the CAS 14, they come with another good version, and they call it AlphaFold 2, or I mean AlphaFold version 2. And this time they release their code. And the fortunate part, or the lucky part, I would say, in the same time, the globe was captured by the COVID-19. And luckily, they also gave their code for AF2. They released the code of AF2. And it was a kind of, I would say, a blockbuster news for the scientific community. And people like me, you, everyone has started working with COVID-19 stuffs and using the AlphaFold too. And no wonder this was real news in 2018, because we saw in 2020 and 21, 22, and still we are seeing that the AlphaFold is producing wonders, and their accuracy level is much higher as compared to Rosetta and iTaser and all those stuffs. Okay, another thing, they use the concept of neural networks, and they they predicted the protein structure, they predicted the protein folding with sufficient accuracy, and that made the job easier for for many scientists. Now let me tell you that why we are always talking about the protein structure prediction and why we are not talking about its real structures. So just for my audience, protein structure is predicted by three methods: NMR, X-ray crystallography, and cryo-EM or cryo-electron microscopy. But these things has severe, it takes a lot of time. This technology is very expensive. And if you see all the structure present in the PDB, they are actually solved by these real methods. I mean, not predicted. NMR, X-ray crystallography, and EM. But there are their own limitations because I am a structural biologist. I know sometime you are unable to to get a good structure, a good crystal structure. Sometimes you are unable to express the protein with reasonable purity or concentration, and if you get it, then the next day, the next challenge is to crystalline it because the crystalline or the science of crystallization is still just kind of a mystery. I mean, just go by a hit and trial method. There is no conceptual things that you will do this so it will be crystallized. No, every protein behaves differently. This is a reality. Okay, and now then electron microscopy came, and if you remember, I think in 2022 or 21, the Nobel Prize went for electron microscopy person. I forgot its name because the electron microscopy does not need the crystallization of a protein. But once again, that is very expensive stuff, and every university or every institute cannot afford it. So, waiting for electron microscopy to solve the structure, and there are millions of structure sequences waiting to be solved. So, it will take ages. So, that's why people are also trying the prediction methods. So, because of the AlphaFold, the COVID-19 era was great. The research was great, and people like they use enormously. They accepted, they grabbed the opportunity of AlphaFold 2 because they released it in 2020. Now, why? You can ask me the question, or you may have a question that if Becker was working in the field of protein structure prediction since long ago than these guys. So why the protein, the half of the Nobel Prize went to these guys for protein structure prediction, not to David Baker? Why AlphaFold? I mean, because AlphaFold accuracy is higher. Point one. The the protein or the protein structure application is broader, and I also told you that it was a COVID time, so it was grabbed by the scientists in both hands. It accepted this gift. It was accepted as a gift, and there were too many thanks to the AlphaFold guys that thank you that you have released the source code, although it was not, it was a commercial company, as I told you, Google's DeepMinds, but they released it. So it has broader applications, people used it, and people received a global impacts because large number of proteins, according to the jumper statement that they were planning for giving the structure of 10 million protein structures. Okay, so that's why, because of this higher accuracy and for this these things, They were the direct competitor of Robita, but they bypassed them, and that's why the international community thought that these guys were phenomenal and it should go to them. But on the other hand, they gave to David Baker the protein design because this concept was brought by the Professor David Baker, and um, if there is any improvement or there will be any improvement, so the mastermind or the person who laid the foundation of this technology is by far, no doubt, there is no one else I can see, is David Baker and his team. So that's why the good news is that David Baker at last received some appreciation in the form of Nobel Prize. And personally, as a structural biologist, I'm very happy for all the three and good wishes to him. And for you, just as those who are listening me, a short message for you that it doesn't matter you are working in microbiology or you're working in mathematics or you're working in physics, whatever you are working. If you don't like your work, stop it. Just start your work according to your interest. Doesn't matter what the people will say that you are a biochemist, why you are working there. No, you can see, you can excel in the field you like. Professor Baker is a biochemist. By nature, he is not a computational, he don't have a computational biochemistry certificate, but for his passion, he continued, he developed his groups, and he did so much thing. The same goes for these guys. Sabi was some game designer, and Jumper was a theoretical chemistry, although Jumper, it's all kind of Jumper field, Professor Jumper. But for Hasabi's, he was not in the rank or not in the field or not known to the biologists till 2009, and but because of his contributions to the field of chemistry, to the field of biochemistry, and from where he came, from which corner he came, you all know that because he was a video game designer. So that's why all these three, they stick to their passion for the work they love, and they got rewarded. Okay. Now, finally, to conclude, a story of generation, the work which was started by others, carried by David Baker, and then crossed David Baker by some Google's DeepMind guys, they shared the Nobel Prize with Baker. So, for me, I will still believe that the contribution of David Baker in the field of protein structure prediction um, are not parallel to anyone, that they are by far above. But still, I wish good luck for this new field. Protein design is still new and it's becoming a reality, and hope you all will read about it and will explore this field and may continue your passion if you want. Thank you so much for listening and for watching this kind of descriptive or historical video. If you have any question, please drop in the comment section. Thank you. Bye bye.